Yeah, Dr. CP is recording it.
Yeah. I mean, we are so happy to have you here, Dr. Becky, madam. I'm really, yeah. I'm really happy that you came in such a short notice.
Now, see, I don't even know how I've talked to you, Dr. CP.
Yeah.
Dr. Jacob John, sir, we are so happy to see you.
Hi, Becky.
Hi, and there's Victor here.
Hi.
How are you keeping?
I'm keeping fine.
Yeah, we have another 2 seconds, maybe 15 seconds for
Our patient manage switch.
व्हिडिओ नाही मला बघायचं नाही त्या लोकांना आवाज येतोय काय
Hello, TV Padmanabhan sir, good evening.
Hi, Sulfi.
Yes, sir.
Hey.
Understand, sir.
Good evening, respected leaders and dear friends. Good evening to one and all from IMA Trivandrum branch. Dear friends, as President of IMA Trivandrum branch, let me to inaugurate AMS chapter activities of IMA Trivandrum and the CME on COVID vaccine prospects and problems. The meeting is called to order. Flag salutation. I welcome Dr. Harihar Sharma for flag salutation. Harihar Sharma.
Yeah, am I audible?
Yes, please.
We, the members of Indian Medical Association, stand here to salute our national flag. Its honor and glory shall be our light and strength. We pledge our allegiance to accredited members of the we swear we will dedicate everything in our power to see it the community of nations.
Thank you, Harihar Sharma. Now let me invite Doctor. I am muting all. Please, the speakers, please unmute yourself and talk. Let me invite Dr. Vasudevan S, Chairman, IMA AMS Trivandrum, to deliver the welcome address. Vasudevan sir.
No, doctor.
Vasudevan sir, can you hear me? There is a technical problem. I invite Dr. CB Kurian, our branch secretary, to offer welcome address.
Thank you, dear President Dr. Prasanth. On behalf of IMA Trivandrum, I welcome our dear National Secretary, Dr. Asokan sir, who on just of the request of our President Dr. Prasanth has offered to attend this meeting and be the chief guest. So, and next I would like to welcome Dr. NARENDRA SAINI, who was the past secretary and now in the charge of Antibiotic Council of the Indian Medical Association and also in the Council of Delhi Medical Association. And third, I would like to invite our world-renowned international vaccinologist and virologist, Dr. Jacob John, sir, who has happily accepted our invitation to share his precious knowledge about the COVID vaccines. We are really honored, sir, to have you on stage. And just now, our capacity has gone to 437, 439, 440, and we have only a capacity of 500. I am not sure whether we'll be able to accommodate everyone over here. So it's just a record path-breaking thing, and it's just because of your magnanimity and your greatness that all of the people have gathered to hear your vital speech, sir. And next, I would like to invite all the leaders, all the national leaders and state leaders, Dr. Alex Franklin, sir, then Dr. Sujadan, sir, and all other leaders. I'm not able to pinpoint each and every one. I would also like to invite the district chairman of Trivandrum, Dr. Binoy, and all other branch leaders, branch presidents, branch secretaries, and all the leaders from all over Kerala. And I would like to especially thank all the MSN leaders who have joined here, and all the students and all the members of IMA Trivandrum branch who have joined here. So I hope you'll have a great, informative and mind-blowing session by Dr. Jacob John. Over to you, Prashant.
Thank you, dear Dr. CB. Next is the presidential address. My dear respected leaders of Indian Medical Association at the national and the state level, particularly our honorary Secretary General, Dr. RV ASOKAN sir, Dr. NARENDRA SAINI, who is past HSG as well as the current chairman of AMR Committee for National IMA, and our beloved, renowned vaccinologist as well as the virologist, our beloved Dr. Jacob John sir, and my dear friends. IMA Thiruvananthapuram is actually really proud. This is a very proud moment for IMA Thiruvananthapuram, and it's the inauguration of its AMS chapter. Dear friends, as you all aware, AMS is the Academy of Sciences, a chapter of specialties. Those doctors, those members who are already enrolled in various specialties of modern medicine, and IMA Trivandrum is committed as one of the largest branches of IMA in the nation. IMA Trivandrum is committed to bring quality training, in the academic sector for the Medical Association. So that's why we have begun with a big bang of bringing a great leader in virology for the most relevant topic. I am not taking your time because today is for academics. Dear friends, at the outset, let me thank our vibrant secretary, Dr. CB Kurian, who has put a lot of pain, and many other leaders of IMA Thiruvananthapuram branch who have put a lot of pain in organizing this event as a grand success. And dear friends, let me invite with a lot of pride and honor our beloved National Secretary General, a great leader of Indian Medical Association, Dr. RV ASOKAN sir, to kindly inaugurate the Academy of Medical Sciences chapter of IMA Thiruvananthapuram and CME. Thank you so much. Ashokan sir, please unmute yourself and talk. Ashokan sir. Ashokan sir was actually traveling, and he was in airport. There was now he seems missing, and Dr. Rajan Sharma sir is calling. Please. Ashokan sir could not join, and he's joining with us in a second time, within a second. Please bear with us.
Dr. Prashant, maybe you can call the next person to address. Meanwhile, he will join.
Sure.
I think Vasudevan sir is there. Vasudevan sir is live.
No problem. Now let us call our chair, Dr. Narendra Saini sir, for his address. Over to you, sir. Narendra Saini sir.
Thank you. Thanks, Dr. Prashant, for inviting me here. And I also thank Dr. SIBY and Dr. Vasudevan, Chairman of IMS. First of all, I'd like to congratulate all of you that so many people are seeing this broadcast. Is a really remarkable thing. We all know that at present we all are disturbed by this COVID-19. Now, this is being infectious disease. India is still struggling with infectious disease. 60% of our diseases are infectious in nature. Now, here in infectious disease, the most important interaction is happening. Dr. Jacob will agree with me, agent, host, and environment. And now, whenever we try to control infectious diseases, one is allowing it its natural course, and in natural course, it will die. But in that process, many people will die. And we have seen in many pandemic, this is happening, whether it is SARS now, which is happening now, or we can solve cholera and Spanish flu. So many people have died, but then natural course, bringing a herd immunity, then is a specific treatment to that agent, which can be virus, bacteria, that agent. But unfortunately, in this COVID, we do not have any specific treatment. So specific antiviral drug which can kill that. So now the third and foremost important thing happens in control is environment, in which we all are seeing that use mask, having a distance, maintaining a distance. But the important aspect in any control is a vaccination. And vaccination is not so an easy thing. Almost it takes 5 to 10 years to form a vaccine of any particular disease. But in COVID, the whole world is working towards that vaccination. And first, the problems comes in vaccinations are first to find a candidate. What can be the candidate for the vaccination, which will start the immunity process? Then is the trial, which goes into phase 1, phase 2, phase 3 trials. Then another important aspect is the manufacturing of a vaccine. Once you manufacture of a vaccine, then it is the transport. It's also a very big issue to be handled. And then giving the vaccination, because at one go you cannot have so many vaccines. So we have to prioritize which people to give, which not to give, and how to prioritize that, which one will be given first one. And so this is itself, all these are big chapters. And this is an issue which is at present disturbing the whole world, and whole world is actually working on that. So vaccination is a big issue. It's not a very small that we just have heard that, okay, polio vaccine you give and things will move on. But once you go into that details, it really is a big task. And to tell us about what are the challenges in this vaccine prospect, and how to mitigate them. I will not say problems. Problems doesn't give solutions, but I'll say the challenges and how to mitigate those challenges. I think we have a very renowned person. I have heard him a lot about Dr. Jacob John, who is a former head of a clinical virology and microbiology, Christian Medical College, Vellore. And he was been a president of our Indian Association of Medical Microbiologists, credited with, the virology work has never been very popular in India when I was doing MD microbiology. But because of people like Jacob, this has become a really pioneer work in virology he has done. So in exemplary work he has done in the field of virology and epidemiology, and he is a recipient of B.C. Roy Award and B.R. Ambedkar Sanitary Award. And I think he is the most, who is the most suitable person to talk on this, the problems or the challenges which you are going to face in the vaccination process. So over to you, sir.
So thank you very much, Narendra Saini sir. Our Ashokan sir has joined. Ashokan sir. Ashokan sir. RV ASOKAN sir. Ashokan sir. Ashokan sir is just joining and he's Ashokan sir, can you hear me? Ashokan sir, please unmute yourself and talk, sir. Ashokan sir, can you hear me?
Prashant, സാറിനെ കിട്ടിയില്ലെങ്കിൽ
He's with us. He's with us. He is actually going to enter into a flight. Rajendra Sharma sir just talked to me.
I think meanwhile, can we have our AMS chairman, Dr. Vasudevan Poti?
Please, for a few words.
Vasudevan sir, are you there? Vasudevan Poti sir, are you there?
Yes.
Sir, please say a few words, sir.
It's a great beginning. You've got an excellent authority on a subject which everybody is actually waiting to hear. How many vaccines are there? What is the importance of each vaccine? What are the challenges in its distribution? Protection each vaccine will offer. These are all questions which everybody is waiting to hear. So it is very great and appropriate that such a talk.
Sir, actually we are waiting for Ashokan sir. Sir, meeting is waiting. Is he in, sir?
So thank you, sir, for having talked to us. And
Oh, sure, sir. Okay, sir. Thank you, sir. Oh, sir. Okay, so Ashokan sir will be coming inside. Now let us go to our beloved speaker, Dr. Jacob John sir. Sir, are you ready, sir? Jacob John sir. Jacob John sir, good evening, sir. Are you ready?
I'm ready.
Sir, your sound is not heard.
How about now?
Better, sir.
Audible?
Better, sir, but not enough.
Okay.
Dr. Prashant.
Yes.
Can you
please tell everyone to mute themselves.
Mute others.
Please.
No, we are sir, muting all.
Sir, Prashant, please mute others. We are not able to hear the speaker.
Jacob John sir, please unmute. Please unmute your again, sir.
I'm unmuted, but somebody is muting me again.
Oh, sorry, sir.
Okay, now
now it's better, sir. You please start, sir.
Prashanth, Dr. CB Kurian Philip, Dr. Asokan, Dr. Narendra Singh, and my dear friends. Perhaps you don't know this, that I'm a life member of IMA. And I joined IMA when I was a pediatrician. For a number of years, I was a pediatrician. And then because of the need in the country for clinical virology, I taught myself virology, did a PhD in virology, and established a virus lab in CMC Vellore. And that's how people got to know me as a virologist. So remember, I'm a fake virologist. I'm a true pediatrician. Anyway, it's my pleasure and privilege to address you in Trivandrum group, because Trivandrum is my second home. I'm an alumnus of Trivandrum Medical College. And I keep in touch with the Kerala government for various things. And just yesterday, the government has invited me to be the chairman of the Kerala Science Congress this time, 33rd Science Congress. I'll be the chairman. It's a great pleasure for me to be getting involved with work in Kerala. I'm also chairing the committee that designed the Advanced Virology Center in Tonnakkal in Trivandrum. So I'm in regular touch with folks in Trivandrum. So it's my pleasure to be with you today. The topic for this evening is prospects and problems of COVID vaccines. Now, there are many viruses that enter through our nostrils. Measles virus, chickenpox, poliomyelitis, respiratory syncytial virus, influenza virus, and this coronavirus. They all enter through the nostrils, get a foothold on the mucosa of the upper respiratory tract. And thereafter, each one of these viruses has a very unique host-parasite relationship. The host-parasite relationship determines both pathology and immune response. Influenza virus, for example, remains mostly in the respiratory mucosa. Does not invade the body tissues to any significant manner. This coronavirus, officially called SARS coronavirus type 2, is also an agent that infects the respiratory mucosa, but then it goes beyond. It invades the tissue, invades the tissues, and causes pathology through various mechanisms. As Narendra Singh pointed out, we do need a vaccine because that's the only way we can protect the life and health of people who are predisposed to severe disease and high mortality, either due to age or due to comorbidities. We do need a vaccine. But let's not pin our hopes on the vaccine as if it would come to our help to stop the pandemic, stop the epidemic, or to help us overcome this particular crisis. Don't pin hopes on the vaccine. We should practice the non-pharmacological interventions like 100% mask wearing, reasonable physical distancing. If everybody wears a mask, then 2 feet between two people is more than enough. Hand hygiene, not crowding around, etc. Be careful in closed space, air-conditioned space, etc. You all know that. Let me tell you that I admire the health management of Kerala because the case fatality rate in Kerala has been for a long time less than 0.5, hovering around 0.46 to 0.5, which is the lowest in the entire country of all the states. And that is mainly due to the work of all the doctors, both in public sector and in private sector, IMA members, and all of you in Kerala have done a remarkable job in keeping the case fatality rate very low. As for infection, as long as you have to breathe, this virus will get around. It's like flood waters and low-lying areas. The low-lying areas will be filled with flood waters, if not immediately, little later, if not later, much later. So certain equalizing force is at work. So you cannot blame anybody or our laxity in discipline for the continued presence of the virus. The virus is respiratory transmitted and it will get around. Moreover, when the pandemic began, the virus was D614 was the genetic makeup at position 614. It has mutated to 614G, and now 85-90% of all viruses globally belongs to the mutated virus, and that is a far more efficient transmitter. And the second waves in Italy, in Germany, South Korea, in Kerala, in many places, most probably is due to the higher transmissibility of the new strain of virus. I can't call it a strain, new mutant, which is a very, very minor mutation as far as the virus is concerned. And it does not change its antigenic structure. So a vaccine, if it is good enough to any strain anywhere, will be good enough for viruses all over the world. So we have hope for the vaccine, but the vaccine is not going to come anytime soon. Now, the immune system is unique in the body, in that we are endowed with a genetic capacity to distinguish between invading microbes, which are also cells, and especially bacteria, and our own cells, or viruses consisting of proteins and genetic material. We have the ability to distinguish between such invading microbes and their structural proteins, and the immune system is capable of responding to them. First with innate immunity, which passes on the message to the adaptive immunity, and adaptive immunity has essentially two arms, the antibody mediated and the cell mediated. The uniqueness of each virus interaction with the body cell is that some of these viruses, for example, measles or polio, antibody is the predominant protective factor. So once you have infection with the measles virus, or with measles vaccine, or with the polio virus, or an immunizing experience with the polio vaccine, you are then protected for life. That is the strength of the antibody-mediated immunity for certain viruses. But that is not true for all viruses. For influenza virus, for example, though antibody is effective in protection, the incubation period of influenza is 1 to 2 days. And the infection, as I said, is essentially on the mucosal surface. So when the virus is inoculated onto the mucosal surface, only 2 days before which you must mount an immune response and have antibodies transduced passively or actively onto the nasal mucosa. There is not enough time. So therefore, you have to keep the immune system always producing sufficient amounts of antibody so that antibody is always present on the mucosal surface as and when the virus enters. That's the protection. That is why influenza vaccination is necessary for everybody on an annual basis. Antibody level should be kept high all the time. And that is the principle for tetanus protection also. You need sufficient tetanus antibody in the body all the time, anytime when tetanus spots are inoculated, because the incubation period is extremely short. So pre-formed high level of antibody is necessary. We don't know enough about the coronavirus. It some ways behaves like influenza virus, and in some ways it behaves very much like a systemic infectious virus. So we are not quite sure. But what we know today is the antibody tests that we have today, which detects antibodies to the famous spike protein, S1 or S2, both are parts of the spike protein, or its receptor binding domain, or the nucleocapsid N antigen. All these antibodies seem to be short-lived, very strange. For measles or polio, for most other things, antibodies don't disappear very fast. But here, in 6 months' time, antibody levels drop to almost undetectable level in the majority of infected people. So, antibody screening has very limited value. And we also think that antibody response has limited value in protection against disease if you are exposed to the virus again. Now, that's not the whole story, because millions of people have been infected, but only less than 1 dozen reinfections have been documented. That means, other than antibody, there must be another protective factor. And we know now today that the T-cell mediated immunity is far more strong against coronavirus than against many other viruses. And it also seems to be long-lived. Why am I saying this? Because if you are going to make a vaccine that has to mimic nature in inducing protective immunity, then that vaccine must induce not only virus neutralizing antibody, which is essentially the spike protein and the receptor binding domain protein, but in addition, T-cell immunity also must be induced by the vaccine. So in the past, when vaccine trials were being conducted with many viruses, all they needed to look for was antibody. But here, you have to now look for antibody and cell-mediated immunity. Both are important. Now, as you all have read in the news media, there are several candidate vaccines that are front runners. The interesting thing about the coronavirus vaccines is that within less than 1 year of detecting the virus, we have vaccines ready almost for licensing in the countries of their origin. How was it possible to move so fast? And that is a very interesting trick in molecular virology.
Jacob John, sir, I am so sorry to intervene. Sir, are you sharing your slide, sir? Many
No,
people.
no slide.
No problem. Okay, sir.
No slide.
Please come
No
closer
slide.
to the mic and say, sir. No problem.
At
Thank you.
night with slides, people will go to sleep. So no slides.
Okay, sir. Please come closer and speak, sir. Thank you.
Those who have questions, please put it in the chat box, and CV Kurian Philip, please manage it so that at the end of my talk, if there are questions, I can answer them.
Sure, sir.
Okay,
Thank
so
you.
how did they manage fast tracking of vaccines now in phase 3 trial, and some of them already measured the protective efficacy and come up with fantastic protective efficacy of higher than 90%. How did they manage it? The vaccines that have come up to that level are not vaccines that we are used to. Vaccines against polio or measles or chickenpox or bacterial vaccines, mumps, rubella, human papillomavirus. All of them have used the classical pathway of introducing either live attenuated or inactivated virus itself, or a subunit in such a way that it is highly immunogenic. To make those antigens, it's a very long process, expensive to produce, purify. It takes long time to bring it to the human use. There are some technologies that were available in experimental laboratories with no vaccine, virtually no vaccine, having gone through these methodologies. And what are they? One is messenger RNA vaccine. The other is adenovirus vectored DNA vaccine. And these are technologies that have never been used in the past, any vaccines. Now let me tell you why these are interesting way to introduce a vaccine. In fact, these are not vaccines at all. These are introducing viral genome that codes for the spike protein. And of course, the virus is RNA virus. Make its complementary DNA, introduce it to the human cells. Human cells is now faced with DNA inside the cells, and that DNA will guide messenger RNA production, and that messenger RNA will go into the ribosomes and produce the protein that we want, the antigen. So the human cells are used as the vaccine producing factories, and by this process, we have two advantages. One is that manufacturing of the vaccine candidate is relatively easy and rapid. Secondly, since the body itself makes the antigen, it is pure antigen. You don't have to purify the antigen because you have already supplied only the genes for the spike protein. And if you inject genes, it is not the RNA gene of the virus, but it is transcribed into its complementary DNA. If you inject that DNA, also body cells may take the DNA in and produce the necessary viral antigen. Sidus Cadila, the Indian company, is going through that route. They are called DNA vaccines. DNA vaccines are not highly efficient usually, whereas if you package it in a vector virus, the virus infects the cells, and the cells now get the virus inside the cells, and along with the virus, the DNA that you put in the virus also gets in. So that is why the virus is called a vector virus. The Oxford AstraZeneca vaccine that the Serum Institute of India is making under the trade name Covishield uses that technique. Chimpanzee adenovirus type 5 is used because you don't want to give a human adenovirus in case it causes some problem in some people. Not only is it chimpanzee adenovirus, which is essentially non-pathogenic for humans, but it is also disabled genetically, genetic engineering, so that the virus behaves like a live virus until it is taken in by host cells. Thereafter, the virus is not capable of reproducing itself. Whereas the genes that were put into the virus, the gene for the spike protein is now becoming active. That will induce the production of the antigen, and then the immune system responds to that antigen. That is the technique of both the Covishield or the Oxford vaccine, and that's also the method that the Sputnik V Russian vaccine has used. The Russian vaccine uses human adenovirus, which is a little more efficient in infecting cells. That also is disabled from replication. So it is harmless, at least theoretically. It has been proven to be harmless. The Russians were very careful in giving adenovirus 26 as the prime dose and adenovirus type 5 as the boosting dose. Now you can understand the reason. You don't want immunity to the first vector virus interfering with the second dose. You're giving one vaccine for stimulating immune response, second dose for different virus for boosting the antibody response, antibody and cell-mediated immunity. So the Sputnik vaccine, the Russians claim, has about 92% efficacy. The two messenger RNA vaccines of Pfizer and Moderna have claimed that they are better than 94, or one of them 95, the other 90.5. So all of them seem to be highly efficient, about 90% vaccine efficacy for the short term against disease, especially against severe disease. The question is, if we need revaccination in future, like we need it for influenza vaccine, influenza prevention, will these vaccines be sufficient for revaccination? We do not know. We have to wait and see whether it will work. Why am I raising this doubt? You would have heard that the AstraZeneca Oxford vaccine seemed to do better, unconfirmed, but preliminary results seem to indicate that it works better if only half dose is given priming dose, and full dose is given as the boosting dose. Now, nobody has explained why this advantage of half dose, full dose, far better than full dose, 2 doses, using the same vector virus. Is the immune response to the vector virus made the first time when you give the vaccine, inhibiting, muting the cell entry of the vector virus when it is given a second time, and therefore is the dose of the second exposure far lower than what you need to stimulate immunity? That's a question that we need answers for, and it will take some time. They are just beginning to do a trial with half dose, full dose in the United States. So we will have to wait and see. So the point is, you will have to wait and see if all these are really true. The regulatory agencies have to look at all the data and then make up their minds whether they are truly safe and truly effective for licensing the vaccine in their countries of origin, and then only it has a chance to be approved in other countries. Now, the classical way of making vaccine is to inactivate the virus. That's what Bharat Biotech has done. The candidate is called Covaxin. It's an inactivated virus. It takes time to grow up the virus and then inactivate it with chemical. And they were smart enough to get an adjuvant, an aluminum salt tagged with a protein, which is a clever idea. It is an idea of an Indian scientist working in Kansas in the United States. And if you link the two together, that adjuvant promotes stimulation of both Th1 and Th2 immune responses. Both cell-mediated immunity and antibody-mediated immunity are enhanced by this particular adjuvant. If only aluminum salt is used, like we use in DPT vaccine, etc., it skews the response to only antibody-mediated response, not much cell-mediated immunity response. So it would have been a mistake to use aluminum salt as an adjuvant for an inactivated virus vaccine, because it would have produced a lot of antibody response, but not much cell-mediated immune response. So they were clever enough to latch on to this new, the very brand new product of the new adjuvant. And so that is what Bharat Biotech is using. So much for the vaccines. So these vaccines are getting ready for registration. Perhaps the mRNA vaccines may get emergency registration for use in healthcare personnel, senior citizens, those with diabetes, etc., those who are at high risk of severe disease and mortality. Protection delayed in them is protection denied for them. So we need to move very fast. So we hope that something will happen before Christmas time, before the end of the year, in the United States, in getting at least 1 or 2 vaccines registered for emergency use, and then it may be saving a lot of lives, because it seems to be highly effective, at least the 2 mRNA vaccines. mRNA vaccines are not suitable at the moment in India for several reasons. One of the mRNA vaccines require minus 70 degrees for storage. The minus 70 freezers are available in many research labs, but they're not available in the cold chain system. The Moderna vaccine seems to withstand minus 20. That is available in our cold chain system. So it may be suitable for India. It can be kept at refrigerated temperature for at least a month and at room temperature for a few days. So that will fit into our cold chain system in India. But those two vaccines are frightfully expensive. To make the mRNA and package it in liposomes, that is fat globules, they will not tell you the trade secret of how they make the fat globules, but the principle is well known. The vaccine product is expensive to make, but very fast to manufacture. So there is hope in the United States. Things may move very fast, but it's not going to move very fast in India. The likely vaccines that might get registered in India, first, it may be Sputnik, because Sputnik seems to be the front runner in India, is undergoing phase 2 and phase 3 trials, and the Russian data are good, Indian data are satisfactory, that may get registered. The Covishield is unlikely to get registered anytime soon because of the problems of the Oxford AstraZeneca vaccine. It is undergoing now a lot of questions. Why is the vaccine's efficacy only 62% when 2 full doses are given? Whereas if you give half dose followed by full dose, which was a mistake in the trial, a serendipitous mistake in the trial, then only the efficacy went up to 90%. And that 90% efficacy is in a particular age group below 55 and in a fairly small group of some 3,000 individuals. So we'll wait and see. The Bharat Biotech's Covaxin will take at least another 3 to 4 months before it is ready for evaluation and registration. So there is hope, but there is no realism to expect a vaccine in India within 2020, and therefore we cannot let our guard down in all non-pharmacological interventions to slow down the transmission and to reduce mortality in vulnerable people. Now, when the vaccine is available, what is the purpose of using the vaccine? Are there more than one purpose in using the vaccine? Of course, yes. The first purpose of using a vaccine is to protect the individual from disease and death. That is preventive medicine. Vaccinated individual is protected. The unvaccinated individual is not protected. However, if you want to control a disease, reduce the burden of disease in the community, then you need a vaccination program. When you give a large proportion of individuals a vaccine, it disturbs the transmission dynamics of that agent, such that not only the vaccinated segment of the population has lower disease burden, the unvaccinated segment of the population also has some indirect temporary protection because the transmission chains are slow and not putting unvaccinated people at risk, like they were at risk earlier. The combined effect of the immunity in the vaccinated and the indirect protection in the unvaccinated, what we call the herd effect of a vaccination program, reduces the burden of disease. That is the visible reduction in burden. The invisible reduction is that the infection burden is reduced. That is a community control of infection. Does the government want to use COVID vaccine when it is available for community control of the disease and the infection, which is much better than individual protection alone? Now, the government of India has announced their priority hierarchy. They have said the hierarchy, the most highest priority will be for healthcare workers, second priority for those about 65, third priority for those between 55 and 65, next priority for people with comorbidities. So this way, you are extending the preventive medicine approach for selected members of the community. It will not amount to community control. Don't you want to reopen schools without fear of transmission? So don't you want to vaccinate teachers and children? Or don't you want to control the infection frequency so that everybody can go back to reasonably relaxed lifestyle, cultural activities, political activities have anyway started. Religious activities can all start if we are in the control mode with vaccine. The extreme control is elimination of transmission. India can lead the world in targeting for elimination of infection if India wants to. But then that kind of a vaccination design has to be thought through. And I don't think that anybody in Delhi is capable of thinking through what it will take to eliminate the virus. Now, as I have just now indicated, the age spectrum that require vaccination goes from childhood to senior citizens. We don't have a platform of vaccinating them. Our available platform is Universal Immunization Program, which is catering to the needs of children below 5 and pregnant women. That will not be sufficient for the coronavirus vaccine if we target for either broadened age group. For example, if you want to give those above 65 and those with diabetes, how do you identify them? How do you give them vaccine? The next question is, will vaccine be available in the private sector? Mind you, the 2 or 3 vaccine manufacturing companies in India, all over the world, are private companies. So the private companies will manufacture the vaccine, and I don't think once it is registered, government can restrain them from selling it into the private market, unless the government uses high-handed technique and prohibits the sale of vaccine in the private market. So more than likely, vaccines will be available in the private market. So then the question is, in the private market, will it be at cost price or cost price with minimum profit, or will there be exorbitant profit that the companies want? Will the public sector distribution of vaccine be free of cost? Now, Bihar election manifesto said, if one party comes into power, we will give it free of cost. Pondicherry government has announced that they will buy the vaccine using the state budget and distribute vaccine free of cost. So these are questions that have not been answered. Then the question is, when will you give the vaccine if it is being given to the age groups higher than children? Schools are not a good idea. We have already seen the school-based vaccination program. There are problems. Schools are not places for giving vaccines. So schools and parents would not be happy for schools being used. But even if you use the schools, it's only a restricted age spectrum. So we have to create a new modality of distributing vaccines. Will it be through healthcare system, or will it be through, for example, ration shops or panchayat offices, or will they use private healthcare system? Because when you give a vaccine, it has got to be under medical supervision. People who get the vaccine must stay around for at least half an hour, 1 hour for any immediate reaction. All adverse events will have to be both monitored, managed means diagnosed and treated. All these are highly medicalized interventions. So you cannot use the vaccine as if it was a food product, safe, you can distribute it. No, it has got to be very carefully given, under careful supervision, under great precautionary steps taken, and all data carefully documented and saved in computers with the mobile numbers of people who take the vaccine, so that they can be contacted, and a follow-up study for measuring both the benefits and the risks on long term. These are the issues of a vaccination program if India moves that direction. So, in short, we have a formidable challenge in front of us. Us meaning the government and the healthcare system. We also have a great opportunity that if we design a coronavirus elimination program and show that its transmission can be interrupted in the whole country, it will be an eye-opener, an inspiration for the rest of the world, giving them the idea that this coronavirus can be eradicated. If we are smart enough, we can cooperate. If the whole world can come together under the WHO banner, the coronavirus pandemic has made sure that the world is completely divided and broken. You have to bring them back. And if you bring them back, eradication of the coronavirus is possible, because we will have many vaccines. Even if we have to give repetitive vaccination doses, then the inactivated virus vaccine or the messenger RNA vaccine, there are different vaccines that will cater to the needs of vaccination for eradication. Thank you all very much for a patient hearing, and I will be happy to take any questions if you have.
Thank you very much, sir. There are a lot of questions. Sir, shall I ask one by one? We have copied from the chat box.
Okay, you select the questions that.
Sure, sir. Sir, actually the vaccination in people taking immunosuppressants like daily, like post-liver transplants. What is your advice?
Yes, that's a good question. They both need the vaccines, but these are non-replicating, non-infectious vaccines, particularly the messenger RNA vaccine and the inactivated virus vaccines or the DNA vaccines would be, you can expect them to be quite safe. When it comes to the adenovirus vaccines, it has got to be proven to be safe. However, since we have a spectrum of vaccines available, immunosuppressed people also can be vaccinated. They may require additional doses of vaccine to make sure that they respond. So from safety point of view, we don't have too much to worry. No live vaccine can be given to immunosuppressed people, but then there are many vaccines that contains non-replicative antigens.
Thank you, sir. Are there different strains of coronaviruses? Is it possible to have reinfection? What is the cause of reinfection? Can the virus mutate and become
that's a
immune
good
to the
question.
upcoming vaccine?
Now, influenza virus, the antigen structure by repeated mutations shifts a little bit. And so the immunity that you had from the previous strain of the virus is not good enough for the new strain of the virus. That process is called antigenic drifting. Antigenic drift.
Thank you, sir.
But in coronavirus, there is no antigenic drift so far. I mentioned one mutation, D to G at 614. That has not resulted in any antigenic shift. So as far as antigenicity and vaccine protection is concerned, the whole world is dealing with one single strain of the coronavirus. So no problem.
Thank you, sir. We all know the effect of prion disease, how it was recognized very late and was due to genetic material. Can mRNA have potential to cause lethal tissues? Have we exhaustively studied them? Are they completely safe?
Good question. As I said first, the messenger RNA vaccine or the adenovirus vector vaccines have never been used in human beings in the past. One vaccine has been used, and that's against Ebola virus, using another virus, vector virus called vesicular stomatitis virus. And that seems to be safe and is highly effective. So we think or hope that this vaccine would be safe enough. But there are people who say that these are genetically modified products, and be careful because you do not know what is likely to happen if you feed new messenger RNA into our cells. What will happen? Well, these messenger RNAs are getting into the cells anyway with coronavirus infection. Okay? Because these messenger RNAs are necessary for viral replication. These messenger RNAs that we're giving are only for the spike protein, but the virus introduces many more messenger RNAs. So it cannot be any worse than asymptomatic infection with coronavirus. So we hope, but we have to have proof over a period of time that these genetically engineered products are safe enough.
Thank you, sir. Sir, the next question is for regarding COVID infected patients. When to vaccinate? Any need of vaccination? Antibody screening, is it useful for them?
Good question. We do not know the answer to that question. As I said, millions of people got infected, but reinfection has been very rare. All respiratory pathogens, not necessarily viruses that enter through the respiratory tract, but then causes systemic disease. For example, measles, chickenpox, polio. One infection is sufficient for lifelong protection against that particular virus. On the other hand, respiratory infections like common cold, flu we have heard, respiratory syncytial virus, almost all respiratory viruses do have second infection, third infection, fourth infection. Repeated infections is quite well known with respiratory viruses. Will this coronavirus act as a respiratory virus or as a systemic virus, which it is? We do not know now. We think that the cell-mediated immunity will function for long-term protection, although we are seeing that the antibody levels decline very fast, just like other respiratory infections. So we are not quite sure. We'll have to wait and see.
Thank you, sir. What all candidates have presented the phase 3 trial reports, and is it any one of them peer-reviewed?
I didn't get the question fully.
Sir, I shall say again, sir. Sure, sir. What all candidates of vaccines have presented the phase 3 trial reports, and is it any one of them peer-reviewed?
Okay. Phase 3 trials. First of all, who evaluates the results of the phase 3 trial? There is something called a data monitoring board, data and safety monitoring board. The phase 3 trial is placebo-controlled, double-blind. Nobody knows who got the vaccine, who got the placebo. After a certain number of the target disease is detected in the whole group, the DSMB breaks the code and determines how many people in the placebo group and how many people in the vaccine group got disease. And overwhelmingly, the deceased people were in the placebo group, both for the Pfizer mRNA vaccine and the Moderna mRNA vaccine. That's why their efficacy is 95% almost. Now, the Sputnik vaccine, we do not know exactly. The Russians claim 92.5% efficacy. If you believe that, yes, that's another vaccine available. These are at the DSMB level. That is not good enough. The regulatory agency, FDA in the USA, the European regulatory agency, India has India's own Drugs Controller General of India, the regulatory agency. They will have to put their own experts to look at the entire protocol of the phase 3 trial and the results and the interpretation and scrutinize whether the safety record is accurate and the efficacy record is accurate and they are believable. And so the regulatory agency will make an independent evaluation for registering the vaccine. Now, the problem is India has India's own registration body, Russia has its own, China has its own, each country has its own. People's trust in them is not as good as the two FDA in the United States and the European regulatory agency in Europe. You can close your eyes and believe them. They will not be biased in terms of making a decision. You know the Russian story. The regulatory agency was willing to register the vaccine even before phase 3 trial was completed, but then they said it's a temporary registration. Now, eventually, all of these will have to appear in the peer-reviewed publication system for the countries outside the countries of origin to have enough trust in the vaccines. So these are parallel processes. Peer-reviewed publication should not delay registration. And these days, of course, you know that the peer-reviewed publication will be fast-tracked if it is about a vaccine, particularly if it is about successful vaccine. So all these are independent elements. DSMB, drug company, the trial people, the drug regulatory control, the agency in the country. So the regulatory agency alone is empowered to license the vaccine. So once that is done, you can believe the vaccine has been adequately evaluated for safety and efficacy. Peer-reviewed publication is icing on the cake for trust in the scientific community. You cannot wait for trust in the scientific community to be an element in licensing the vaccine, because, as I said, protection delayed is protection denied to the people who get severe disease and die.
Thank you, sir. Next question is, human adenoviral vector vaccines like Sputnik, will it be effective in India, where majority might be having antibodies against adenovirus, which might clear the vector?
That needs to be investigated. The Sputnik uses human adeno 26 and human adeno 5. These are not very common adenoviruses. These are not the fever and sore throat producing adenoviruses. So therefore, it's quite possible that they will work everywhere. But when it comes to India, we have to check whether we have antibodies against these. Supposing you have very high antibody level against adenovirus 5 or 26, human adenoviruses, then when you inject the virus, the antibodies will neutralize the infectivity of the virus, and the virus will not enter host cells. Therefore, there will be no DNA producing the spike protein. So you have to wait and see. The reason why they chose 26 and 5 is because they are the least prevalent adenoviruses. And the reason why Oxford used chimpanzee adenovirus is that nobody has any immunity to the chimpanzee adenovirus.
Thank you, sir. Next question is the vaccine safe in pregnancy?
Now, that you could guess that vaccine should be safe because these are non-replicating antigens or vaccines. However, guess is not good enough. It will have to be tested, and testing will have to be done extremely carefully. First of all, in all the phase 3 trials, if women were involved, and if anybody was found to be pregnant later on, perhaps unknown to them at the time of volunteering to get the vaccine, those data will have to be culled out by the DSMB. And therefore, all phase 3 candidates, particularly female, will have to be kept under surveillance or under observation for a long period of time. The other thing is, in animal models, you have to give to pregnant animals and see whether there is any transmission of anything to the fetuses. The human placenta is very unusual, therefore we have to look at placenta and see whether either the vehicle adenovirus will not pass the placenta. We know that, or at least we had no data that they cross the placenta. But what about the fat globules that we carry the messenger RNA? The bottom line is we need to prove that it is safe in pregnancy, it's safe in young children, etc.
Dr. SIBY, unmute yourself.
Yeah. Sir, there are actually some more questions.
The voice is going low.
Yeah. Sir, can you hear me?
No, SIBY, please louder.
Yeah.
No problem, sir. I shall ask next question.
Yes.
Please, SIBY.
That's okay. Can you hear me now?
Yes.
Yeah. So one question is, are we justified in trying to vaccinate healthy population? Why is natural immunity to other coronaviruses not being considered?
Very good question. Now, there are common cold coronaviruses. There are 4 common cold coronaviruses that just cause coryza. Nobody is worried about them. 2 of them are beta coronaviruses, phylogenetically close to the coronavirus of COVID. 2 of the others are alpha coronaviruses, genetically distant from the beta coronaviruses. Why are they important? Karolinska Institute found that blood donors in Sweden, before the pandemic reached Sweden, 25% of the blood donors had cell-mediated immunity against the COVID SARS coronavirus. So where did it come from? Most probably from the common cold coronaviruses. Okay, after the pandemic reached Sweden, the prevalence of cell-mediated immunity rose rapidly because of the coronavirus epidemic. So there are hidden cross-reacting antigens, most probably common cold coronaviruses. Now, the common cold coronaviruses are also, like other respiratory infections, short-lived immunity and repeated infections. Now, if you have repeated infections, then you have stronger immunity. Do overcrowded communities, countries with very high population density, countries with low respiratory hygiene, people spit anywhere, they sneeze without closing their nose and mouth, they cough in buses without closing their mouth. In such communities, will there be repeated coronavirus infection and better immunity? Well, we have not seen any effect of that because the coronavirus infection in
Sir, there's a problem with your voice, sir, breaking. Dear friends, there is a technical problem with the Jacob John sir's connection.